So arguably the next portion of the non-oxidative phase of the pentose phosphate pathway, which our overview of the pathway is shown here. The next phase of this is to take our our current pool of 15 carbons. So if you remember, we have 3 ribulose 5-phosphate molecules. So that's a grand total of 15 carbons. We're able to procure 1 ribose 5-phosphate molecule and 2 xylulose 5-phosphate molecules. What we're going to do is we're going to take 2 of those 5 carbon molecules. So a ribose 5-phosphate and xylulose 5-phosphate molecule. And with those 2 molecules, we're going to do what's known as a transketolase reaction. So the carbons number 1 and 2 on xylulose 5-phosphate are going to be transferred and added onto carbon number 1 of ribose 5-phosphate. Now, the result is going to be a glyceraldehyde 3-phosphate molecule and a acetoheptulose 7-phosphate molecule. At this point, I think that it's fair to wonder, like, what arguably is the point? And one of the points for the pentose phosphate pathway is, I think, best illustrated when you think about glyceraldehyde 3-phosphate. Where have we talked about glyceraldehyde 3-phosphate before? Well, glyceraldehyde 3-phosphate, or GAP, or GA3P, is talked about as one of the products of the aldolase reaction in glycolysis, where you take fructose 1,6-bisphosphate and break it into 2 3-carbon molecules. One is dihydroxyacetone phosphate, and one is glyceraldehyde 3-phosphate. So, with that said, this is a common metabolite. Just as glucose 6-phosphate is a common metabolite between the pentose phosphate pathway and glycolysis. Now, a very good question to ask then is, well, where are these 2 different pathways taking place? And both of these pathways in eukaryotic cells are taking place in the cytosol. So if you were to say that a glyceraldehyde 3-phosphate molecule generated in the pentose phosphate pathway is in the same environment that a glyceraldehyde 3-phosphate molecule generated in glycolysis is, you'd be correct. So you have a little bit of almost crosstalk and proximity between our 2 different pathways. So we're generating glyceraldehyde 3-phosphate, which arguably could find its way into glycolysis. So I think it's reasonable to say, and kind of think about almost prioritization of our glucose molecules and prioritization of our carbon-containing compounds. If you think about glucose, a primary goal would be to send it through glycolysis to get some ATP from it. Well, what if you have, just kind of think about it as you have too much glucose, but not an excessive amount, but you have too much glucose. Well, one thing that you could do is you could take that glucose, phosphorylate it one time, and kind of redirect it towards the pentose phosphate pathway. And when you redirect it, you more or less sequester it away. You can say these carbons are going to be used in this way. They're going to be put to use because we're going to generate some NADPH. We're going to get some glyceraldehyde 3-phosphate, but that's not the end of the road. Ultimately, what we could do in the right circumstances is come back the other way and use that glucose 6, you could regenerate and go back into the pentose phosphate pathway and put those molecules back into glycolysis. So I always kind of think about the pentose phosphate pathway as a pathway that serves its own role, generates NADPH, generates ribose 5-phosphate, but also it's a means of kind of sequestering away carbons from glycolysis. And we see that, or we see kind of another illustration of that with this common metabolite of glyceraldehyde 3-phosphate. Now, what I think is really important to do is to think about how many total carbons do you have between these 2 compounds, ribose 5-phosphate and xylulose 5-phosphate, which it's helpful to think what's the relationship between those. Well, they are isomers of one another. Well, between these two, we have 10 carbons. Between a 3-carbon glyceraldehyde 3-phosphate molecule and a 7-carbon cetoheptyl 7-phosphate molecule, you have 10 carbons. So we have these two kind of reorganization of our atoms. We're not generating any NADPH, but we are generating something that arguably, that glyceraldehyde 3-phosphate, that could find its way into glycolysis. So this is our second major step in the pentose phosphate, or in the non-oxidative phase of the pentose phosphate pathway. We're going to proceed on in just a moment, but I hope this is helpful.